The diagram below shows the hepatic cycle that captures ammonia and detoxifies it. Boxes A through D represent substances or enzymes either entering or leaving the cycle. Which substrate or enzyme best represents each of the boxes? Box A is fumarate. In the conversion of arginosuccinate to arginine, fumarate is cleaved off. Box B is urea. In the conversion of arginine to ornithine, the R group of arginine is cleaved off as urea. Box C is the enzyme ornithine transcarbamylase, which converts ornithine to citrulline inside the mitochondrial matrix of the liver. Option D is aspartic acid. Aspartic acid condenses with citrulline to form the product arginosuccinate. The definition of BUN is the measurement of urea in the bloodstream. A low BUN value would indicate that the organ, the answer is the liver, is malfunctioning, whereas a high BUN value would indicate the organ, the answer is the kidney, is malfunctioning. How is carbamophosphate synthetase 1 deficiency distinguished from ornithine transcarbamylase deficiency? Carbamophosphate synthetase 1 deficiency is distinguished from OTC deficiency by orotic acid urea. Orotic acid urea would be indicative of OTC deficiency because its substrate, carbamophosphate, would accumulate in the mitochondria matrix of the liver, and when it accumulates, it'll spill into the cytoplasm and enter pyrimidine synthesis, forming the pyrimidine precursor, orotic acid. Shown are the various features of the major genetic disorders associated with amino acids. Fill in the empty cells of the table. PKU, phenylketonuria, is caused by a defective enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine. The enzyme requires the coenzyme tetrahydrobiopterin to carry out the conversion of phenylalanine to tyrosine. When the enzyme is defective, the hallmarks of PKU are mental retardation, microcephaly, and musty odor of urine. Albinism is caused by a defective enzyme tyrosinase. When the enzyme is defective, its substrate tyrosine accumulates and melanin cannot be synthesized. Tyrosinase requires the cofactor copper. A clinical hallmark of albinism is hypopigmentation. Alkaptonuria involves a defective enzyme homogentistate oxidase, which converts tyrosine to homogentistate. The enzyme requires iron to carry out this conversion. When the enzyme is defective, the characteristic of alkaptonuria is dark urine. Also, onchorosis, that's the darkening of the cartilage, can occur. Maple syrup urine disease is caused by defective enzyme branched-chain ketoacid dehydrogenase. This enzyme works on the carbon skeletons of isoleucine, leucine, and valine after the amino group is removed from those 3 amino acids. The enzyme needs 5 different coenzymes: thymine pyrophosphate, lipoic acid, coenzyme A, FAD, and NAD. When the enzyme is defective, 2 clinical hallmarks are mental retardation and maple syrup odor of urine. Isovaleric aciduria is due to a defective enzyme isovaleril-CoA dehydrogenase. This enzyme works downstream of branched-chain ketoacid dehydrogenase by working on the carbon skeleton of leucine. The enzyme needs FAD as a coenzyme. A clinical hallmark is sweaty feet odor. Cystinuria is due to a defective transporter for basic amino acids, such as arginine and lysine, but this transporter also transports cysteine. There is no coenzyme or cofactor needed by the transporter. When the transporter is defective, cysteine will accumulate, forming dimers of cysteine, which are known as cysteine. The problem with cysteine is it's not very water soluble. Hartnup disease is due to a defective transporter for large neutral amino acids, including tryptophan. There is no coenzyme or cofactor needed by the transporter. When the transporter is defective, pellagra-like symptoms occur. diarrhea, dementia, and dermatitis. Cystathionuriria is due to defective enzyme cystathionase. When the enzyme is defective, homocysteine will accumulate, which will then go into making more methionine. The enzyme needs pyridoxal phosphate as a coenzyme. Clinical hallmarks are benign for cystathionuriria. Homocysteinuria is due to defective enzyme cystathionine beta synthase. When the enzyme is defective, homocysteine will accumulate and will back up into making more methionine. The enzyme needs pyridoxal phosphate. When the enzyme is defective, homocysteine will accumulate, giving rise to deep vein thrombosis, dislocated lenses, and stroke. In catecholamine synthesis, tyrosine is converted to dopa by the enzyme tyrosine hydroxylase, which uses tetrahydrobiopterin as a coenzyme. Dopa is converted by the enzyme dopa decarboxylase to form dopamine by using the coenzyme pyridoxal phosphate. Dopamine is converted to norepinephrine by the enzyme dopamine beta hydroxylase, which uses vitamin C and copper as coenzymes and cofactors. Norepinephrine is converted to epinephrine by the enzyme phenylethanolamine N-methyltransferase using S-adenosylmethionine as a substrate. In the treatment of Parkinson's disease, why is carbidopa often used together with the administration of levodopa? The answer is Parkinson's disease involves the loss of dopaminergic neurons in the substantia nigra. While dopamine cannot cross the blood-brain barrier, levodopa does cross, but is converted relatively quickly to dopamine prior to reaching the barrier. Carbidopa does not cross the barrier and increases the levodopa effect by inhibiting peripheral metabolism of levodopa. Pheochromocytoma is a tumor of the adrenal gland and is characterized by excessive synthesis of catecholamines, resulting in hypertension and weight loss. Lead affects heme synthesis by affecting the enzyme ferrochelatase, which is located in the mitochondrial matrix of a cell. The effect of lead on the Km of the substrate for this enzyme is increased, and the effect of lead on the Vmax of this enzyme is decreased. Lead poisoning results in accumulation of a substance that can be used diagnostically, and this substance is zinc protoporphyrin 9. Briefly discuss the biochemical rationale for the most common treatment of lead poisoning. Pharmacological chelators, such as deferoxamine, would bind to lead, and the lead chelator complex would eventually be eliminated in the urine. The decision to fortify a widely consumed food with an essential nutrient is often politically and medically controversial. For each of these two proposed fortifications shown below, briefly state one medical reason in favor of it and one medical reason against it. Regarding the fortification of flour with iron, a reason to do it is because anemia due to iron deficiency is a common problem. On the other hand, a reason against it is because of hemochromatosis, iron overload, is a common problem. Regarding the fortification of flour with folate, a reason to do it is by increasing folate intake that would prevent neural tube defects in pregnancy as well as decrease megaloblastic anemia problems. A reason against it is by increasing folate intake that can mask anemias caused by B12 deficiency. Bacterial flora carry out a number of reactions that have a direct bearing on human metabolism. Concisely describe the action of the flora in the following. A. Metabolism of a specific water-soluble vitamin. The bacterial flora synthesize vitamin B12 and biotin. B. Metabolism of a specific fat-soluble vitamin. The bacterial flora synthesize vitamin K. C. Bile acid metabolism. The bacterial flora deconjugate glyco and taurine-containing bile acids. In other words, they remove the amino acid glycine or taurine from bile acids that are conjugated with those two amino acids. D. Cholesterol digestion. The bacteria have no effect. E. Carbohydrate digestion. Cellulose and other fibers are partially digested. The bacteria undergo anaerobic fermentation and by doing so result in bloating and flatulence. F. Bilirubin metabolism. The bacterial flora can deconjugate bilirubin diglucuronide and by doing so they reduce bilirubin to the urobilinogens. A 13-year-old boy slid into home plate while playing baseball, sustaining a large bruise on the thigh that was initially dark red. The boy was sent to the hospital where x-rays were negative, but the bruise became dark blue-green in color. After a few days, the parents took the boy back to the doctor with the concern that the affected area had become a yellow-orange color. The physician calmed the parents by explaining the events in simple layman's terms. Concisely explain the events in biochemical terms. In a bruise, red blood cells are being turned over. The heme portion of hemoglobin is a red-purple pigment. The heme undergoes conversion to biliverdin, which is a green pigment. Biliverdin gets further oxidized to bilirubin, which is an orange-yellow pigment, causing the color changes in a bruise. A 2-year-old girl was taken to a pediatric clinic because of persistent anemia and failure to thrive. Laboratory analysis revealed blood levels of vitamin B6 to be extremely low. Would anemia be expected to be pernicious, hemolytic, megaloblastic, or microcytic? Circle the best answer. Concisely explain the biochemical relationship between your answer and vitamin B6. The anemia would be expected to be microcytic, and the biochemical reason is because vitamin B6 is needed to make the coenzyme pyridoxal phosphate. Pyridoxal phosphate is needed by the enzyme ALA synthase in heme synthesis to convert ALA to heme. If there's a deficiency of vitamin B6, heme cannot be synthesized, therefore the red blood cells will not develop properly and will be small in size.